So hello everyone. Yeah, it's an honor for me to present with the great support of Jessica and Srijit these few slides about giving you an update about this OASIS consortium, exploring cell painting and omics for liver toxicity assessment. So can you click, Connie? So some background information to tell you about this, and this was a little bit mentioned at the beginning of this annual meeting. We are typically in a cross-sector initiative where we try to make use of the context and the questions we are facing in the AgChem sector and the one in pharma with regard to reduction of animal testing. So overall, the question can be seen as very similar, but there are some subtle differences because in our case, in the agrochemical sector, there is strong push to go up to the point that did happen 10 years ago or so with the cosmetic industry about the total ban of animal testing, which is not the case for the pharmaceutical compound. If you click on Connie, there is one animation. So the idea for this is to, and the vision is to make a multi-sector, a multidisciplinary collaborative effort, serving both the specific needs of AgChem and pharma, and to generate useful insight in the context of new approach methodologies. Next slide. So OASIS, in terms of mission, the mission is to gain confidence in the combination of in vitro methods using different types of readouts, so cell painting, transcriptomics, proteomics, in the context of safety assessment. And to make it something reasonable, we decided to focus on one type of toxicity, hepatotoxicity, as a use case, being also having in mind that it was a compromise between the interest from the agrochemical sector and the one from the pharma. So the objective is to benchmark a rather large set of chemicals for which we have at hand in vivo hepatotoxicity profiles, both in rodent and in human from clinical trials, and to make these translations and to make this understanding and these learnings from the cell-based assay using molecular and phenotyping readouts. Next slide. In numbers, OASIS is rather big thing with 16 industry, 15 academic labs, 7 governments, and 2 non-profit partners. Hired 2 amazing postdocs, Jessica and Srijit, who will be presenting together with me. We have also 1 PhD student, Mariam, at Grégorian at Université Côte d'Azur in France. I'm co-mentoring with Professor Vincent Van de Waal. We launched 4 work streams dedicated to pushing the consortium forward. So one on compound selection and data extraction, big thing, study design, QVIV, quantitative in vitro to in vivo extrapolation, and the whole thing about data management and the integrated data analysis to make the magic out of the data that's going to be generated within OASIS. So far, we completed the search in 5 public databases for compounds that have associated rat or human in vivo data, and also a lot of work with regard to the coordination of compound procurement, because we need not only the in vivo result, obviously, but also a little bit of powder to make the experimental part being possible. Up to now, we compiled a list of 1,300 compounds. The plan is to test at least in 3 cell types. Next slide. So this is currently the list of the current members. I won't go into all the details. So it's the list of the different types of partners from academia, governments, and industry, and the non-profit. Next. Alors, from an overall and the overview, the global overview about the objective of this project and the vision is to, as I said, make use of as large as possible set of public and private compound with associated in vitro and in vivo data in rodent or in human. Test them in different cellular cell models. Gain some knowledge about the effects those molecules are inducing in vitro in those different cell types using cell painting, transcriptomics, and ELISA for proteomic profiling. And out of those data generated, make a comprehensive analysis to make all the conceptual learnings we can make from the translation from in vitro to in vivo, having also some estimate of exposure of this regarding these molecules in order to make all the learnings, as I said. So the project is meant to be fully pre-competitive. All the data are going to be made available in the public domain at the end of the project, and there will be also the publications and both from the data and from the methodologies. Next slide. This is the classical parallelogram, a little bit twisted in a sense that we start with this about this translation from rat cell culture, in vivo rat data, and also from human cell culture and human clinical data, and make these translations and these learnings across those different, what I would call spaces. And going maybe in that direction on the next slide, another way to present the translations we want to achieve is about what we could call the spaces we want to explore. So if we go along the continuum of different spaces covering, we can cover when it comes to the safety assessment of chemicals, we can start from the left-hand side with the chemical space. Then we move on the in vitro biological spaces, then in vivo biological space in lab animals, and then in vivo biological space in human. And all those different kind of spaces, which are represented to these two-dimensional representations, are different in nature. So if we go into the chemical space, where in this two-dimensional representation, one dot represent one molecules, you will have distinct and overlaps also spaces between the drug and agrochemical sector, or even some industrial chemicals. When it comes to in vitro biological spaces, this will depend on the test system, so the species derived for which the cells are derived, the cell types, the time of exposure, the concentration, and the readouts. And when it comes to the in vivo spaces, you will have, of course, the species, if it is a rat, human, or whatsoever, the genetic variations, the sex, the tissues, the time, the dose, and readouts. On the next slide, in OASIS, the intention is to cover all those spaces which are highlighted in yellow, from the chemical to the in vitro biological space in human. And more importantly, if you click one more, Connie, make the learnings and the interplay between all those different spaces. Just to give you one example, is about how much the response one can obtain in some tissues in the rat, so in the liver rat, can compare to the one in human. Or to give another example, how much it is important to look into a primary cell culture in a given species compared to a cell line, or using different cell types. So let's say osteosarcoma cell line, to predict, for example, or to make the learnings for the liver. So in a nutshell, those are the different spaces within OASIS we intend to cover and to better understand their interplay among themselves. Next slide. So now we are moving on to the work streams updates, and I think it's now the time for Srijit to describe with the couple of slides coming ahead.
Yeah, so we have been collecting compounds from last year from different sources, including Drug Matrix, TGGate, StoxRepDB, and various other sources, looking at those that we have proper stereochemistry data, tautomer non-chimerization, etc. So we ended up with a large list of compounds after block listing from the industry, around 1,490 compounds, and of which we also developed a prioritized subset along with Jess helped do that. To find 541 compounds for which there was some kind of in vivo data or pre-existing rat repeated dose data in 30 days, 90 days studies, etc. So these two are available on the website already. And let's go to the next list. Yeah, so we just go to the next one as well, maybe study design. Yeah, so for now we are we have drafted a cell painting protocol which has been used in the jump CP dataset, and we are trying to set up the plates with different controls and trying to find out where to place those, optimizing the plate batches based on where to place the compound, such we don't get any batch effects because we're repeating experiments multiple times. And we have completed asset development at the Broad with U2OS cell lines. HEPA-RG is in progress, and then the primary rat hepatocytes would happen next year. So should we go to the next slide? Yeah, let's go to the next one again. So for the QIVIVE, we have downloaded in vitro and in vivo parameters that we could for benchmarking different experimental data sets. We are using predictive models like Opera from NIH and PK Smart, which I developed, and then SimPlus, which is an external test set, external model across all these chemicals to generate these PK parameters and comparing these predictions with experimental data, trying to find whether we can derive ranges for confidence for each parameter. We are going to use the HTK ICE IVIVE tool to generate conversion factors for the Cmax and CSS. There are different models there for PK, PBK, like 1 compartment, 3 compartment modeling to find the oral dosing, etc. And then using these QIVIV models, we will be able to generate some kind of PK parameters and in vitro to in vivo extrapolation that we can compare whether the animal data we have can make an informed decision about human toxicity. So this is the proposed workflow for the next year. Let's go to the next slide. Yeah. So the way the compound selection, yeah, let's stay here. The way the compound selection happened was we gathered 1,400 compounds in the public domain last year, and we are getting another 200 from industry partners, which are different from the public domain compounds. Once we have these two sets of compounds, you saw 1,500 something compounds that you currently already have, and then there's another 200 coming in. We'd probably end up having around 1,700 compounds, of which we would be able to order most of them, hopefully, and be able to run them across different omics pipelines with different cell lines. And so the goal is to have a matrix of these phenotypes across different compounds, and then trying to do a QIVIVE, which I explained before, but also integrating the data across different phenotypes, trying to predict liver toxicity data that we have, or even find out which particular cell morphology markers or which particular gene expressions can actually give a clear answer about liver toxicity in a particular cell line. So there's a lot of exploratory work to be done here next year, which is really exciting. So let's go to the next slide again. Yeah, so currently we have finalized the compound lists. The study design is happening right now. We are collaborating with EU OpenScreen and Fenaros in Europe, who are doing HepG2 screening as well. We have already had Axiom, which is one of our partners here, give us some data on human hepatocytes, which we don't cover in OASIS. So that's also some really complementary data that Jess is analyzing right now. And we have been able to do a lot of image profiling using Cell Profiler, but also state-of-the-art deep neural networks and convolutional neural networks that give us embeddings for these images. So there's a lot of different types of data that we have had access to right now for the cell painting side, and it's really exciting to have transcriptomics and protein profiling data coming up soon. So maybe David or Jess take over from here if you have anything else to add.
Jess, yeah.
Yeah, I think we can go to the next slide. Yeah, so I'm really excited I get to present the first data that's come in. So just to kind of give some background, Axiom is a startup. They're trying to develop predictive models for toxicity using large imaging data sets. And they've actually collected images from primary human hepatocytes for over 50,000 different compounds that they've screened. And they very generously made the compounds that overlap with the OASIS list publicly available. And so we have those available online, and we are analyzing them now. So this data set that they've made available contains images for 1,085 compounds at 8 different concentrations with 2 replicates each. These are screened in a pool of primary human hepatocytes from 5 different donors that are mixed together. A really amazing thing about this data set is that they're also collecting biochemical assays that are aimed at understanding cytotoxicity, and these are paired at the well level. So for every single little group of cells that we have images of, we also have the MTT and LDH assay results. This data set is also collected at 40 times magnification. It's more normal for people to collect at 20 times. And in this 40 times magnification, I have one little screenshot right here. That stuff in red is the mitochondria. And so you can make out at really high resolution that mitochondrial matrix. I did want to give a little bit of background just on what the cell painting assay is, just in case there are people on the call who haven't encountered this data type before. So for cell painting studies, what we typically do is grow cells in a 384 well plate. And in each different well, we would add a different perturbation. So that would be DMSO or some concentration of a compound. Next, all of the cells are washed with inexpensive dyes that stick to different parts of the inside of the cell. So for example, the mitochondria or the nucleus. And then we take pictures of them with a microscope. And so here's a very simple example with just 2 channels. Normally we have 5 or 6 showing the nucleus and various parts of the cytoplasm. Next, we put it through a computational pipeline where we segment each of the cells and even various compartments within the cells, for example, separating the nuclei and the cytoplasm. And then for each of these identified cells, we'll measure thousands of features. So the most common one is to use Cell Profiler, human designed or defined features that measure things like the shape or the size or the intensity of the different fluorescent channels within each individual cell. But we can also get profiles for these cells using deep learning methods. So all of this comes up with a large data set where we have measurements at the single cell level for not very much money, so you can collect really large data sets. Next slide, please. So the first thing that we did with this data was do concentration response analysis so that we could calculate the in vitro point of departure. I'm showing here the results for the cell profiler features from the cell painting data sets. And I used both the global and the categorical Mahalanobis distance method for summarizing each of these profiles as kind of a single score that we can use to fit curves to get an overall general bioactivity point of departure. We also fit curves to the MTT assay results, the cell counts, and then the LDH assay, where those later 3 are all assessing cytotoxicity in some way. So MTT measures the metabolic capacity of a group of cells. So if the cells are generally have impaired function, the metabolic capacity will go down. And also if you have fewer cells, there's going to be a lower metabolic capacity. LDH is an enzyme that's released when membranes are ruptured. So it's generally a marker of really catastrophic cell death. So what we see right away is that the cell painting morphology detects bioactivities for more compounds and at lower concentrations than the cytotoxicity assays. So we see a cell painting based point of departure for 56% of compounds. Next, we have the MTT, which makes sense because impairing the metabolic phenotype or metabolic capacity is a little bit more subtle of a phenotype than outright cell death. Then we see cell count, and then we see LDH, which is kind of this most extreme form of cell death. Next slide. So another thing that we're doing beyond just fitting curves to everything is starting to do supervised analysis of these cell painting profiles. So since we have this paired cytotoxicity assay readout data, as well as the cell painting profiles, we can build supervised models to try and predict the assay readouts from the cell painting features. So here's an example I'm showing you predicting the MTT assay readouts using cell painting and the XGBoost regression model. And then in that little snapshot right there, I just have a comparison of the random null. So that's quite different. And we see an R squared of 0.74. And just for some background here, since we can also just compare MTT to just the overall cell count. And there we only get an R squared of 0.16. So we see that there's really quite a lot of information in these images that's allowing us to predict this much more subtle phenotype than just outright cell death. We have many more results than the ones that we have time to present here, but another thing that we're doing is going through ToxCast and taking all of the chemicals that are in both ToxCast and that we've measured in OASIS, and we're trying to build predictive models for whether a tested compound in this OASIS list was a hit in the various ToxCast assays. And then as well as trying to use RefChemDB to evaluate some of the predictions that we've made. Finally, we're also comparing the performance across lots of different factors. One is just the relevance of the experimental design. So when we're going through all of these ToxCast assays, we're stratifying these by cell type and also the type of outcome that they're measuring, with the idea being that something that's measured in a cell-free assay or in like a breast cancer cell type is going to be much, much harder to predict from data measured in primary human hepatocytes than assays that were measured in a liver-relevant cell model. And we're trying to show, we see very, very good agreement between our cell painting data and the assays that were measured in parallel. So we want to understand, how that predictive performance declines as we get into assays that are measured in very different experimental context. When we're building the predictive models for the ToxCast assay hits, we're exploring different ways of focusing on specific concentrations in the OASIS data. So for example, one of our hypotheses is that if we filter out non-bioactive concentrations and cytotoxic concentrations, that will be more predictive of specific assay hits than if we use all of the concentrations. And then finally, Srijit mentioned this already, we're looking at different what we call cell representations. So I explained already the cell profiler method, which is where, we put it through the software that measures all of these human-defined features. But these days, deep learning methods are getting extremely good at turning an image into a set of numbers that can be analyzed quantitatively. And so we're benchmarking about 3 different deep learning methods that have been either trained on images of cells or even these deep learning networks created by groups like Meta AI that were trained on large data sets of just cats and dogs and buildings and stuff. And we can use all of these to get quantitative profiles of cells. So we're evaluating how well these profiles work compared to the ones that we're used to seeing from Cell Profiler. Okay, next slide. All right, so next I'll just close up with some various presentations that we've done. We've been at a number of conferences over this year. A lot of these were Srijit. A lot of you have probably seen him around presenting posters on the OASIS project. You can go to the next slide. All right, so for 2025, we have plans to complete the cell exposures. We've already developed the data analysis pipeline for the cell painting data, but we'll modify this for the transcriptomics data and the targeted protein data. We'll continue to share the results and especially the data with consortium members, and then generate an OASIS compound library for others to purchase, because one of the things that we found in this project is that a lot of people want to run this list, in their own assay that they're developing. And so the purchasing has been like a sticky point. So trying to streamline this and make this easier. And then finally plan all of the future phases of the work. Next slide. All right, I think that's all. So we probably have time for some questions.
Yeah, really nice job. I'll just leave on the 2025 plans. So if folks have questions, feel free to unmute or you can put in the chat. Really nice overview from the 3 of you. Thanks for walking through that. See some claps. Oh, there, Falcon has a question. Go ahead. Oh, I saw Falcon for a second. Oh, yeah, there you are.
it was my mistake, but really good progress and really good work. So thank you both.
Okay, great. Scott, was that the same for you? Were you trying to clap? And
yes, I was trying to clap.
okay, let's go to Jan Willem then.
Thank you. It's a very nice presentation and a very good study. So is it correct that you use 8 concentration levels for each compound? And for me, it's not fully clear because of the details, but what is the shape of the dose of the concentration response curve? Is it I'm a bit afraid that you will derive just one concentration as a no effect level, but I would suggest to try to have enzyme kinetics or pharmacological response analysis on that 8 concentration level. That it should have a lot of promises in that way to have a detailed dose response curve.
Yeah, I mean, we're not so much calculating the no effect level, but more, fitting a whole curve to all of the responses using the approach outlined by the NTP, and that's implemented in the BMD Express software. So we're fitting 8 different parametric models to each of these concentration response curves, and then trying to calculate this benchmark dose according to the variability that we see in the control conditions.
Mm-hmm.
But yeah, we're doing this separately for each compound, right? So each of these is going to be shaped differently.
Okay, thank you. I'm interested to see the results.
If I may just add one or two sentences that just in addition to what you just said, Jess, is indeed the idea is to evaluate the dose response and get some sense of the dose response concordance according to the different technologies. So cell painting is one technology. How much the dose response concordance is similar to the one of transcriptomics or the one from proteomics is also part of the question, and how much this thing compare to the dose response we can see from in animal testing. That's going to be also one of the key learnings we want to try to make in OASIS.
Thanks, David. See a couple hands. Let's go to Jing next.
Hi, very nice to talk. Can I ask about how do you define bioactivity on your, I think, slide 97? So I'm just wondering, it's because you got so many compounds in this study, can you start to give like a compound-specific bioactivity rather than like just we can see LTT signal, LDH signal. So can we get more from the cell painting study?
Yeah, so that's when we're analyzing these images and we're coming up with quantitative features that describe each cell, we're measuring so many different things that we hope that it gives us like a high content coverage of a wide area of biological space, similar to what you'd capture with a whole transcriptome measurement, although we're looking at like the shape of different cell components. And so when we do this bioactivity dose response analysis, we're really computing the overall distance between each profile from the controls. And so if we see a perturbation at this really high global level, we're just calling this a change in bioactivity, but really this means that we're surveying a huge space. So I should have made this more clear when I presented this slide, but we're really excited about these results because since we see cell painting much lower than even these sensitive indicators of cell function like MTT, that's kind of our hypothesis that this kind of unbiased high content readout is capturing like a lot of sensitive mode of action signals that are happening at lower concentrations than impairment to cell function.
Great. Thanks, Jess. Thanks, Jing. I do see 2 more questions. Let's do those both, but then we will have to move on. So let's do Magnus next.
Yeah, hello. I'm talking from Sweden, so it's at night here, well, almost. But I have a question about the pipeline you talked about, where you have cell profiler at the end, but you also talked about the deep learning approaches. And what is the progress there, and what is your prognosis regarding the best approach? Is it either or both these approaches would be suitable for the next step, so to speak?
Yeah, I have not even presented this, I think, to Connie yet, so this might be new results for her. But what I've been finding with this is I'm just using deep learning to get the features for the cells. I'm using the entirely identical post-processing and curve fitting and prediction pipeline afterwards. So you really just see the impact of using this deep learning approach to get the features. One of the things I notice is in all of the supervised prediction analyses that I'm doing, the features are performing the same. So that makes me feel like they have similar information content. But in some of the other results, like in the curve fitting, I actually see more consistent and better results for the deep learning methods. And I think that's because deep learning methods create features that are nicely normally distributed. And so they just behave better in some of these calculations like Mahalanobis distance computation. So I don't think it's a question of information content. I think the deep learning features are just a little bit nicer to work with. But then the downside is they don't have names, they just have numbers. So it's hard to kind of interpret what they mean.
Yeah, thanks.
Thanks, Magnus. Peter?
Hi, gorgeous talk. So it's really encouraging to see, the extra hits that you see with cell painting. And so because you have so many compounds, are you going to try to overlay some of the cell painting data onto, say, chemical space to see if there's hotspots that sort of coincide between the, although you're sort of agnostic, you don't know what actually it means in terms of, your deep learning output with chemical space to sort of try to make predictions as to chemically how these compounds are working?
So I think like another thing that the Carpenter Singh Lab has done is collated large databases with like the various targets, like protein targets of many different compounds. And so we can use all of that information to kind of quantitatively analyze the trends that we see. So one of the things with that MTT prediction plot I showed you, there's a whole bunch that are not predicted well. And what's really interesting is when we do an enrichment analysis of what targets those are hitting, those are mainly hitting xenobiotic transporters. So we wonder if it's a case where the cell looks like it should be able to metabolize, but it's just not because that substrate is not there because it's getting transported out or something like that. This is all speculation right now. But I think maybe you're asking more about like a more QSAR approach, and I would have like about actually analyzing, the chemical structure. I'd have to let Srijit answer that because that's not so much my background.
Yeah, and keeping in mind that, there's only around 1,100 compounds here. So there's two levels that we can address the kind of question you're asking. One is a pretty, let's say fuzzy, certain compounds fit into certain classes and will maybe be able to identify that the ones that have this kind of impact on liver have this kind of image-based appearance and they cluster together and so on, like at the annotation level, at the classification level. Whether we can do anything more like a quantitative, are there certain side groups associated with certain image-based features and then with certain liver outcomes? We may not have enough representation for that, but that's where Srijit could advise.
Yeah, I think we need to explore this further and look at different representations and see what we get. I don't have an answer for this yet.
Okay, thanks everyone.
Thanks, Peter. And thanks everyone. And I'm glad we got Anne for at least a little bit of today. Huge thanks to David, Srijit, and Jess for presenting. If folks have more questions, I think they could put in the chat. And even if these folks have to go, I can make sure that gets to them.